I think where things get maybe more exciting and even more impactful is when you can start to apply the formulation design and protocols that go with it to really optimize the overall cell health for an intended drug therapy. And where we are on the timeline for that I shared a little bit already, is that we will have some off-the-shelf formulations can be used in clinical trials starting later this year for a number of cell types. But I actually think that some of the work we're doing on the custom work with specific targeted programs with specific therapies, we will probably hit clinical market with formulation on those efforts even faster.
Welcome back to the Smart Biotech Scientist. I'm David Brühlmann, and we're continuing our eye-opening conversation with Jeffrey Allen, who is the chairman and CEO of Evia Bio. In part 1, we explored the revolutionary science behind their osmolyte-based cryopreservation technology. Now we're getting practical. How exactly can you implement these methods in your lab? What specific protocols make the difference between mediocre and exceptional cell recovery? And if you're developing the next breakthrough cell therapy, how will these techniques impact your entire supply chain? Whether you're commuting to the lab or taking a quick coffee break, these next minutes could save you months of frustration and millions in development costs. Let's dive back in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. How does your 4-step assessment work? Let's say we look at the freeze-thaw protocol. Is that a theoretical exercise, or would you even do some tests at your facility, or that would be then the client? How does that work?
We can do it both, actually. We do some work at the client site, some work at our site. It depends how comprehensive they want the audit to be. But if they want a totally comprehensive audit, it tends to require us to go to their site so that we can see how they're loading the controlled rate freezer, as an example, and to see how they're handling materials. One thing that we find often is protocol creep. So the protocol is written perfect, but they're not following the protocol. And so we go and we can kind of work with them to understand the different processes they've got in place, the different processes, how well they're following. And then we'll share a comprehensive report at the end of an audit to share with them all the things where they're already adapting best practice and just keep doing what they're doing. And then we'll identify areas where there's opportunities for improvement. And if they want, they can retain us to help them implement some of these corrective actions, although we give them the playbook so that they largely can do it on their own if they want to also.
So where would you see this going ideally, let's say in the next few years? Because you mentioned that you're still in this development state, but very close to a breakthrough. What impact will this have on, let's say, cell therapies, where in particular the logistical challenges are very difficult to manage?
Yeah, it's interesting. The way I see it evolving for both Evia and also for the clients we serve is the service side of what we're doing will be a constant where if somebody really wants to improve their own competencies, call it a tech transfer of process, if you will, we're going to have that capability always, and we believe that there will be a need for that always. I think where things get maybe more exciting and even more impactful is when you can start to apply the formulation design and protocols that go with it to really optimize the overall cell health for an intended drug therapy. And where we are on the timeline for that I shared a little bit already, is that we will have some off-the-shelf formulations can be used in clinical trials starting later this year for a number of cell types. But I actually think that some of the work we're doing on the custom work with specific targeted programs with specific therapies, we will probably hit clinical market with formulation on those efforts even faster. And I think once we get to market with those efforts, we'll have published data, published papers that the industry can feed off of to take that into account for their own efforts. And so I do see a big transformation in terms of what the standard and benchmark is for cryomedia formulation and protocols. And I think it'll be a process that takes 2-3 years to really take root. But I think we're well on our way in terms of the early stages right now.
As technologies are improving almost at light speed, we have now more sophisticated algorithms. We have AI, we have machine learning tools, we have hybrid modeling, and so on. The list is endless. To what extent would it be possible in the future to undergo this audit you're doing and pretty much predict the right formulation, the right protocol almost on one click? Or is that maybe too optimistic?
That's pretty optimistic because the combinations are almost endless when you think about the combinations of factors. But I do think that when you leverage the experiences that we've built through so many years in the lab and additional experiences we're now getting on an accelerated basis with all these customers applying even more and more cell types, I do believe that the starting point gets accelerated. And so we forego a lot of the early work that it would take to develop the data behind the algorithm. And so maybe not one click, but I think that you can get there with the equivalent of a handful of clicks. I think we know enough where we have insights for what probably will work with regard to both formulation design and protocol.
Now, this last part of our conversation, I would like to shift gear a bit and focus on your leadership experience, because you have led organizations through significant transformation and growth. And I always love to speak to leaders and just pick their brain a bit. So can you share a specific high-stake decision you made as a biotech leader that paid off? And what was the principle behind?
I'll do my best to share a single example, but I would say that it's got applications pretty much wherever I've been. If I just go back to my first experience in cell therapy with a starting material company, you're talking about 2 founder-led small companies to create a platform. So you've got limited resources and you've got 2 completely different founder scientists, different ways of doing business, different cultures and things like that. And so anytime you come new into an organization, I think it's really important that you do 2 or 3 things. Number 1 is get alignment on strategy. And I love doing strategy development and really pulling in all the thought leaders, all the stakeholders on a team, and effectively doing a brain dump of all of the collective smarts around the table and making sure that you leave no stone unturned with respect to the possibilities. From there, being able as a unit to gain alignment on, okay, given all the bountiful data in front of us, what the strategic possibilities, what are our priorities going to be? In this particular case, with the starting material company, it was really important that we got a unified kind of overall company culture and identity. And it sounds like, well, that's simple, but it's not simple when your founder's identity, that's been their livelihood. It's kind of like their own child. And so to be able to do a marriage of sorts of company leaders and create a unified identity is not as easy as it sounds. But by getting the alignment early on the strategy, the priorities, kind of the company identity, it just makes it a lot easier as you go forward to execute when everybody's on the same page. One other thing I would add to this is I tend to operate with an awful lot of passion. And I love to throw, once we have that alignment, just my passion for the science, for our mission into the mix. And it seems to be often infectious that people start getting excited with me. And the one thing, I'm not just proud of the fact that we grew 6x in 18 months, but I'm equally proud that we did not have a single staff turnover in that time as well. And I think that you want to get smarts, but you also want to get kind of commitment and passion from the team to execute your deliverables. And that's what happened in that case.
This is amazing. Well done. And nevertheless, I want to ask you this last question about your journey. If you could go back to your, let's say, 30-year younger self, what piece of advice would you give that person?
It's interesting because I was kind of almost naturally strong in the financial world and business world. I didn't really think I'd end up in the sciences, and then I ended up working in the sciences. I immediately had this tremendous passion for the sciences, and I had to kind of learn it on the fly across my career. I keep going to these different fields in the life sciences. Each time I go to a new field, I have to learn new science from almost square one. And so I wish I would have developed more scientific roots early before I even jumped into the workforce. But the parallel is I work with all these scientists, founders, entrepreneurs, and they're kind of probably maybe often lacking the one thing I started with, which is the business acumen and the finance acumen. And in my case, it was easy to have a passion for the science and then learn it on the fly. I kind of think that the scientists would to take a little bit more time to learn the business and finance side. But regardless of what any of our strengths are, one pillar of philosophy that I've had with me that's really helped to be successful is I love surrounding myself with really, really talented people. And I'm only going to be so good at so many things, and I'm going to have a lot more gaps than competencies overall. And one thing that's not only been helpful to be successful, but it's actually been rewarding and fun is to learn from others by having a really strong, talented team around me and lean on them for their expertise to help drive the company forward. And so my lessons learned would be to do that even earlier in my career.
Yeah, Smart Biotech Scientist, I'm going to repeat it because this is spot on. Surround yourself with smarter people, learn from other people. And I notice that often as a scientist, especially early in the career, you should not show weakness, but actually go into rooms where you're not the smartest person. That's where you learn.
I used to like to say that if I was the smartest person in the room, we're all in trouble.
Yeah, that's a great way of seeing it. With all what we have covered today, Jeff, what is the most important takeaway?
I would say one size doesn't fit all. And I would say with respect to Evia Bio, we do have an awful lot of expertise to ensure that whatever your drug development program is, whatever cell type you're working with, we can help understand the unique requirements of your program and let us partner with you to optimize for that. Yeah, and I would say that's the biggest single takeaway.
So tell us, Jeff, where can people get a hold of you?
It's jeffrey.allen@eviabio.com. And on LinkedIn, it's actually Jeffrey D. Allen 2 is the URL, I guess you'd call it.
Excellent. Smart Biotech Scientists, do reach out to Jeff. You'll find all the link down in the show note. And Jeff, thank you so much for sharing your passion, your insights. It's been a huge pleasure. Thank you so much for being on the show today.
David, it's been an honor, and I really appreciate the opportunity to get a chat with you today. So thank you.
And there you have it. Practical insights from Jeffrey Allen that you can implement in your lab starting today. From optimizing freeze thaw protocols to rethinking your entire cell therapy supply chain, these strategies could be the difference between a therapy that reaches patients and one that fails in manufacturing. The difference between standard and exceptional cell viability can transform your entire development timeline. Book a free call with me to discover how you can fast-track your biotech development without the costly delays. I'll help you find solutions specifically for your challenges. Visit BrühlmannConsulting.com/call to schedule your consultation today. Until next time, this is David Brühlmann reminding you that when it comes to preserving the cells that could change patients' lives, every percentage point of viability matters. Thank you for listening to the Smart Biotech Scientist, where we make bioprocessing smart, simple, and successful. All right, smart scientists, that's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.